Good day, everyone. Welcome to the STEM Lockdown Digital School. My name is Kadar Matibe, and I'll be presenting the grade 10 life sciences lesson to you guys. Now, guys, remember to use my email address at ls.wisnet@gmail.com for anything related to life sciences, and I will gladly help you guys. So remember, we are focusing on revision. Hello, Ray. So this week is mainly revision, and also next week before our study break. So we are going to have a study break from the 10th of June to the 16th of June. Remember, I told you guys that we are going to have exams. That is why we are doing revision, so that you guys can remember everything that we covered. So from the 17th of June to the 26th of June, we will be assessing you guys. So we'll be having online assessments. On what platform? I am not quite sure as yet, but I will tell you guys as soon as they alert us. But prepare, study, revise, ask questions, use my email address if you are still not sure of any concept. We are also offering extra classes on Saturdays. So if you're still unsure of a topic or a section of a topic, use the time we'll be having on Saturday. So the timetable is on our website. Use that time to ask questions. All right, so that we can go through everything that is still confusing you. So guys, today we are doing revision on the cell structure and function. So the practical part of cells. That is your microscopes, all right, so at the end of this lesson, I want you to be able to answer questions on this topic in tests or exams. So guys, when we give you questions, you must take time, read first those questions, and then you can answer. Don't just rush to answer questions because of time. So take your time, read the question first and understand it, read it again, and then you may attempt to answer it. All right. So yesterday I gave you a homework because we couldn't finish our lesson, and it was on tests for organic and inorganic compounds. So I hope most of you did that. All right, so study the table below, which shows the results of certain tests on 5 different food types. And these food types are food type A, food type B, food type C, food type D, and food type E. So these are the name of tests and color change observed. So for the Benedict's test or Felling's test, color change for food type A was orange, for food type B it was blue, for C orange, for D blue, and for E orange. And the iodine test for food type A was yellow, the result. For B was black. For C it was yellow. For D it was also black, and for E it was black. And lastly, the burette or Milon's test for food type A, the result was pale blue or white. For B, it was pale blue or white. For C, purple or brick red. For D, purple or brick red. And for E, purple or brick red. Now, which food type A, B, C, D, or E contains the following? So which food type contains only starch? So which food type contains only starch? So it means when testing for this food type, like when testing for other organic compounds on this food type, other organic compounds gave us a negative test. And for the iodine or for the starch test, oh guys, I gave you an answer. For the starch test, it tested positive. So Aaron is saying A. Remember the starch test, we use iodine. Nope. Pile, you can say rice because they didn't tell us that it is rice. They only gave us food types in alphabet. Yes, it might be rice because rice is rich in starch, but which food type? A, B, C, D, or E? All right, so you're saying D. So it means other food tests were negative, but for the iodine test, the test was positive. So I only have two options that you guys gave me from Aaron and Pile, which is A and D. What are the rest of you guys saying? Sanel is saying C, food type C. KK Slider, Tyrese, Mila, Musa. What are you guys saying? Ray, Dilo. Ray's also saying C. All right. So the correct answer, unfortunately, is B. Oh, so my B flew up there. All right. So the correct answer is B. Why is it B? Remember the Benedict or Fellings test is used to test for glucose, which is a monosaccharide. And the Benedict solution or the Fellings solution is blue in color. Okay. And the Burette test, the Burette solution is also blue in color. The Milan is white in color. And the iodine solution is brownish in color. And when there's the presence of starch, it changes to black. So because we want a food type which only contains starch, we're going to look at where the iodine test was black. So it means A and C are cancelled out. So those who said A and C are wrong. So you cancel out A and C and you choose from only the three. And it says only starch. So meaning that other solutions which were used to test on these food types had to be negative. And the Benedict solution, because it is blue in color, it remained blue for B. So it means that B or D is the right answer. And the Burette, because it is also blue, it remained blue for B, but then it changed for D, meaning that D might contain proteins. So it's not only starch. So food type D doesn't contain only starch, but it contains starch and proteins. So the answer is B because only B contains starch. So it turned blue black or black because it contains starch. So you guys get that? That's how you read off from this table. All right. So because I gave you guys an answer for B, for A, what is the answer for B? Which food type only contains glucose? Meaning that glucose tested positive and other solutions used became negative or their color didn't change? This one should be easy. Peter says C. A. Aaron says B. Only glucose this time. Sanel says A. Mila A. So I'm getting a few A's. The answer is A. This is because for A, food type A, the Benedict solution changed color from blue to orange. Remember, if there's the presence of glucose, it changes to brick red or orange. And the iodine solution stayed yellow or brown, meaning that it didn't change. And the burette didn't change. Okay, so if the burette and the Milan test changes, you will see a purple or brick red. And if the iodine changes, you will see black. And if the Benedict changes, you will see orange. But if it doesn't change for Benedict, you'll see blue color, iodine yellow, burette pale blue or white. Okay, so for A, food type A only contains glucose and doesn't contain starch or proteins. Okay, so the answer is A. And number C, which food type only contains proteins and glucose? So I gave you guys an answer. So which one contains proteins, and glucose? C, C, C. You guys are getting the hang of it. So the answer is C, because for the Benedict, it changed color to orange. And the burette, because this is the test for proteins, it changed color to purple. But for iodine, it didn't. So food type C contains proteins and glucose. And for D, which food type contains starch, glucose, and protein? So the food type that tested positive for all of these tests, which one was it? Mila says E, Sanele E. All right, so you guys, the two of you are correct. When you look at food type E, for the Benedict, it changed from blue to orange. For iodine, it changed from yellow to black. And for the burette test, it changed from blue to purple or white to brick red, which means that there's a presence of starch, glucose, and proteins. So you guys got that correct. And the last question on this is rice contains starch and proteins, but no glucose. So which food type A, B, C, D, or E is probably rice? So which food type is probably rice? A, B, C, or D? It contains starch and proteins. So it does not have glucose. Aaron is saying B. Which food type tested positive for starch and proteins? B and E. Pila says B. Aaron is saying no, it's D. It tested positive for starch and proteins. Look at the table carefully. Mila says D. So for those who are saying B, just check the table again. So which one didn't we mention here? This is quite obvious. We didn't mention starch and protein. So you look back at the table, and for glucose, it's blue, meaning that it didn't change. For iodine, it's black. And for burette, which is for proteins, and iodine is for starch, it changed. Then it means the answer is D. Nope, Pile. The answer is D. It's not C, because C contains proteins and glucose. So you guys can take a picture of this. Let me go back. Sorry. Take a picture of that and let us do the other one. The other homework. It's also on food tests. So I hope you guys get that. It's kind of confusing, but you guys will get the hang of it. All right, so Ray's asking, how is it D? Let's quickly go back. All right, so it's D because the statement reads that rice contains starch and protein. So it means that rice should test positive for starch and it should test positive for protein. But it does not contain glucose, meaning that it must test negative for glucose. So when we look at D for glucose, the solution should change. If it's positive, then it should be orange. But if it's negative, then it stays blue. So in our case, there's no glucose in rice, then the solution should be blue. And then we head to iodine. So for the iodine test is for starch. And we know that when there's the presence of starch, the iodine solution changes color to black or blue-black. So in this case, for food type D, the iodine changed color to black, meaning that there is starch. So it changed from yellow to black. So black denotes that starch is present. And for proteins, if there are proteins present, then the burette test will change color from blue to purple, or the Millon's test will change, the solution will change from white to brick red if there is protein. So it changed color to purple, meaning that proteins are there, are present in rice. So D is the answer. There is no glucose because glucose tested negative. The solution stayed the same. Yes, Ray. All right, so Tyrese is doing some guesswork, and that is correct. Also, by judging from the previous answers, you can see that B only has starch, A is only glucose, C is protein and glucose, and so on. Therefore, D is basically the only one left, which should contain starch and proteins. But don't do that because we might confuse you or trick you guys by saying which one contains fructose and proteins. Remember, fructose is also a monosaccharide, it's a sugar. So the answer will be the same as C. So do not guess, but you might do that. All right. So I'm glad you guys get that. So 2.3, Elena carried out 4 different food tests to investigate the composition of onions, potatoes, and peanuts. The results are recorded in the table below. So the chemical reagent used was iodine solution, the Benedict's or Fellings, ethanol and water, and burette or Millon's test. So you know that iodine tests for starch, Benedict's for glucose or simple or reducing sugars. Sorry. Ethanol and water tests for lipids. And burette tests for proteins. So the results they got, iodine in onions, it tested negative, in potatoes positive, in peanuts negative. Benedict's positive, in potatoes negative, in peanuts negative. Ethanol and water negative in onions, negative in potatoes, and positive in peanuts. What does it say about ethanol and water? Remember what it tests for? And the burette tested negative in onions, positive in potatoes, and positive in peanuts. So from the above results, identify the type of organic food compound contained in each of the following. So what do we find in onions? Looking at this whole table. So for onions, we find whatever compound that tests positive when using the Benedict solution. So what is it? What is that compound that tests positive? If it is present, that the Benedict solution tests positive is if it is present in a food substance. What is that compound? We were talking about it in the previous slide, so you guys should just give me the answer. So Mila is saying glucose. So glucose is present in onions. And what is present in potatoes? Look at the table for potatoes. So potatoes tested positive, or the iodine solution used tested positive in potatoes, so meaning that something is present. And also the burette, so two compounds are present in potatoes, starch and proteins. Yes. And 2.3.3 for peanuts. What do we find in peanuts? Which compounds? Yes, Pile, starch and also proteins. Because we use the burette to test for proteins. What do we find in peanuts? We find proteins and lipids and proteins, or fats and proteins. Yes, guys. So for peanuts, compounds found there are fats and proteins. And which of the above chemical reagent used did not require heat to show a result in any of the foodstuffs? So which solution used or chemical reagent did not require heat to show a result on a food substance? So which of the four chemical reagents did not require heat for it to show a result? This one is quite easy. It's actually very easy. Aaron says ethanol. All right, so the answer is the iodine solution. Because you just use a few drops of those on, to place drops of that on the potato, and it just turns blue-black immediately because it contains starch. Yes, Ray says, ma'am, by my understanding here, For starch, there is no need to heat the result or to heat. The result is almost instantaneous, but the liquid needs to soak in to dry the solids. That is correct. We don't need heat because the result is almost instantaneous. So we're looking at the result that appears fast, okay? And also ethanol. But then you guys have to wait and shake the whole beaker so that you see the emulsion. And then add water. You shake again the test tube to see the emulsion. So in this case, we are saying it's iodine because the results are almost instantaneous. All right. But also ethanol is correct because alcohol and heat don't go together, guys. They're not the same WhatsApp group because the reaction might catch a light. So you must be careful when using ethanol close to a heat source. All right. So the answer is just iodine solution because of the reason Ray gave us. So state 2 precautions that need to be considered when conducting food tests. What are the precautions that you guys need to consider when you are conducting food tests? All right, so because of time, all right, so the answer is glucose for number 1. Number 2, starch and proteins. Number 3, fats and proteins. So onions only contain glucose. Potatoes contain starch and proteins, and peanuts contain fats and proteins. That is based on the results we got. And the iodine solution did not need a heat source for us to get the results. And the safety precautions are do not heat test tubes over direct flame and use gloves when using the Millon's reagent as it is poisonous. All right, so those are some of the precautions you guys can take whilst doing those practical experiments. Now, to focus on today's work, which is on cells, so microscopes. It's not a lot of work. So if we finish, I'll be happy. All right, so question 1, multiple choice. If the magnifying power of the ocular or eyepiece in a microscope is 10 times and the magnifying power of the objective is 40 times, the total magnification of a specimen will be? All right, so that's awesome. Ray has the previous paper that I am giving you guys. So if you want him to share it with you guys, ask him. All right, so you guys can share. Oh, hey, sorry, Ray. I'm sorry about that. So you may ask her to share with you guys so you can exchange contact or email addresses. Also ask me and I'll send you the paper. All right, so if the magnifying power is for the ocular lens is 10 times and the objective lens is 40 times, the total magnification of a specimen will be A, 10 times, B, 40 times, C, 50 times, or D, 400 times. What is the magnification? So Pila says B, it's 40 times. Or you guys can create a WhatsApp group That way you guys can share homeworks for those who didn't take screenshots, share answers and everything. All right. But be safe because other people are on this platform just to disturb and become nuisances. So you guys be safe when sharing your information. Rather, send it privately to that person. Don't send it there. All right. So Mila says it's D 400 times. So the answer is D. Remember, for the magnification, the total magnification, you multiply the ocular lens or the magnifying power of the ocular lens by the magnifying power of the objective. So this is 10 times multiplied by the 40 times of the objective lens, which gives us 400 times. All right. And the scientist who first called box-like compartments cells is A. Robert Hooke, B. Anton van Leeuwenhoek, C. Charles Darwin, or D. Albert Einstein? Aaron says B. Sanele A. Ray C. We've never mentioned Charles Darwin and we've never mentioned Albert Einstein for the topic of cells. All of you who are saying C and D, y'all are wrong. So it's either A or B. So we only have one A, and the answer is H Musa. Yeah. So the answer is A, it's Robert Hooke. So Robert Hooke was the first scientist who first called box-like compartments cells. Remember, he used Koch cells. He viewed Koch cells under a simple microscope in 1665, and he called what he saw cells. He saw box-like compartments and he named them cells. I said you guys should focus on these two for cells, the two scientists. But it's fine. Moving on. Study the diagram below and answer the questions that follow. So we have this apparatus here. What is this apparatus called? So what is this? What is this apparatus called on this diagram? That is a microscope. So number one, it's a microscope. Provide labels for parts A, C, D, and E. So this is part E, ne? The whole of it. Part A, it is this circular thing here. C, the structure here. D, and E. What are those? What is part A? Remember, microscopes differ. A is the adjustment. Is it the fine or the coarse adjustment, Sanele? The fine or coarse? That is the coarse adjustment. So it is the coarse adjustment. And what is part C? No, part C, not B. So A is the coarse adjustment screw. B is the fine adjustment screw. But what is C? What is C and what is D and what is E? So C is the handle, D is the base, and what is the structure? E. E is where we put our slides on. What do we call that? The stage. So E is the stage. All right. So C is the body tube. All right. So provide functions of the parts labeled A and E, only those two. What is the coarse adjustment screw used for? What is the function of it? And what is the function of part E? I just gave you a function for the stage. So you are more than welcome to take a screenshot and I will give you answers just now. So for number one, that is the microscope or light microscope, like you guys said. A is the coarse adjustment control. C is the arm. D is the stand or full plate or the base. And E is the stage. All right, so what is the function of the parts labeled A and E? All right, so you guys, all right, so A moves body tubes rapidly up or down to focus on the image for A. Oh, okay, so that's the function of A. It moves the body tubes up or down to focus on the image. That is what Aaron is saying. So let me quickly give you the answers because of time. All right, so we place slides on the stage. That is for E. And remember, the stage has clips which hold the slides tightly so that they do not move left or right. All right, so the coarse adjustment screw is used for the initial focus of the object by moving the stage up and down, bringing the object closer to or further away from the objective lens. So Aaron gave us that answer. And E, the microscope slide is placed here. It contains clips to prevent the slide from moving. So those are the functions of the coarse adjustment screw or control and the stage. Now, tabulate 4 differences between the light and the electron microscope. Let me just go back if you didn't get that. And in the meantime, give me 4 differences between the light and the electron microscope. How do these two microscopes differ? Remember, I gave you guys a table the last time. How do they differ? Right, so these are the answers. These are the differences. So the light microscope uses a beam of light. It is small, it is not expensive, and you do not require training in order to use it. You can only see color images, and the specimen can be alive and unharmed, sorry. You can use lower resolving power or lower magnification with the light microscope. But with the electron microscope, it uses a beam of electrons from the name. So you can tell that the electron microscope uses a beam of electrons. It is large and not portable. It is expensive and you require training. So the color of the images is black and white images. The specimen must be dead when you use this type of microscope, and it provides greater resolution and greater magnification. So any four of the following, you get a mark. All right, so you guys can take a picture of that. Remember the differences between these two. You might be asked in the exam. So remember, the exam or the online assessments will be from the 17th of June to the 26th of June. On which platform? We are not quite sure yet. And you guys are going to have a study break from next week, the 10th of June to the 16th. Yeah, to the 16th of June. And then from the 17th, your exams or tests are starting. So you guys better prepare. And on Saturdays, we have extra classes. So you guys are more than welcome to log in, tune in, and ask questions. So what I would prefer you guys to do is send me all the topics you are finding difficulty in for both term 1 and term 2, so that we can focus more on those and prepare you more for your tests and exams. So these are some of the references I used. And in the next lesson, we are going to look at past paper questions for the cell structure and function theory topic. So animal cells, plant cells, cell organelles, tissues, what are organs? That's what we're going to look at tomorrow. All right. So guys, if you have any questions or if you're still unsure and want me to go back to a topic, hit me up on my email address at ls.wisnet@gmail.com. And also follow us on all social media platforms. On YouTube, subscribe to our YouTube channel at Africa 10 Gigs. Follow us on Facebook, Twitter, and Instagram. And also subscribe to our daily newsletter, guys, online on our website at Africa 10 Gigs, okay? Enjoy the rest of the afternoon. See you guys tomorrow, same time, same place. And thank you for participating and for being online. I really appreciate it. Stay safe, keep warm, wear masks, sanitize. See you guys. Goodbye.